Hello everybody and welcome to the first video of 2021. I'm sure that we all hope that 2021 will be better than 2020, and I would say that it can go worse, but never say never. So let's try to be positive and let's try to start the year at best. So this webinar hasn't been planned much and it's a little bit random, I must admit. Just one day I thought, why not recording a webinar and sharing some cytology cases with you, because I know that you like when we share cytology cases. So the only theme that there is in this webinar is cytology of fluids. So I chose a specific group of lesions of cytology samples, but except for that, they're pretty much random. I call it afternoon tea for two reasons. One, because, as I said, it's a random chat, a random selection of cases, and also because I live in the UK, so you know that the afternoon tea is a very traditional kind of experience, quite joyful, quite relaxing, and I hope the webinar is going to be the same, but also because I want to give you the idea of something quite fun, quite friendly, and I'm sure that I'm not the only one that follow a lot of YouTube channels and find quite entertaining and relaxing sometimes to watch video of people that maybe they don't do cytology, they do something completely different. And so I thought, why not doing the same? So you might be at work, you might be at home having dinner, or maybe having a tea. I'm having a tea right now, that's why I call it afternoon tea. And this can be a way, yes, to learn something, of course, but also to relax. And it's also a way of interacting with you. I know many of you personally, I don't know personally, but I know by email, by message, and I think that this is a way, a nice way to, in a way, keep in contact and creating a relation. So let's get started. And before we start, I just have a couple of general considerations that I think are important, in particular for those that are not very familiar with cytology. When I was looking at the people that agreed to join the chat, I've seen a lot of experienced people that probably, I mean, I don't know what they're going to get from this, hopefully some fun anyway, but I'm sure that there are a lot of people that don't know much about cytology. So I thought just in a couple of minutes, I want to give a few very, very basic information. So when we talk about cavitary effusion, we are essentially referring to three types of fluids. So the pleural fluid, the pericardial fluid, and the peritoneal fluid. Obviously, this is just a small part of the fluids that we can see on cytology, because we can obviously sometimes receive samples from other fluids like tracheal wash, bronchoalveolar lavage, synovial fluids, or urine. But I would like to focus now on cavitary effusions. Now, we're not going to talk about sample collection, we're not going to talk about sample preparation, but we are going to focus on sample analysis. And as I said, we are going to do it with some cases. If you want to know more about this topic, that I think is quite interesting, and it's a kind of topic that involves, of course, clinical pathology, but also involves other disciplines, in particular internal medicine. I will give you some other ideas at the end of the talk. And when we think about the classification of effusion, this is the classification that for a very long time, and still now for many clinical pathologists, has been used. So the classification into transudate, modified transudate, and exudate. And this classification is mainly based on the nucleated cell count of the fluid, which can be performed manually or more often with automated analyzer, mainly the hematology analyzers, and also based on total protein, which can be measured with a biochemistry analyzer, or can also be estimated with other technique, like with the use of refractometer. Obviously, each of these fluid has a different cell composition when we look at them under the microscope. So, for example, transudates, usually they have a prevalence of mononuclear cells, whereas exudate, usually they have a prevalence of neutrophils. However, this classification, it's not used as much as it was in the past, because there are quite a lot of limitations. One limitation is that it doesn't include, it doesn't mention other fluids, other specific fluids that we can encounter, like, for example, chylus, uroperitoneum, or biliary effusion, or neoplastic effusions, which often they have high cell count and high protein, so they might look and appear like exudate, but they are actually not exudate. But the reason why this classification is not in use anymore, or not much, is because of this concept of modified transudate. And if you look into the human medicine, you will see that this concept, this term modified transudate, it has never been used, never been in use. Whereas in veterinary medicine, it's something that it has been used for a long time, and still we see it in many pathology reports. And by looking into the literature and I wanted to find out where this term came from, I noticed that this term has been introduced for the first time in a book, in a veterinary clinical pathology book in the 60s, to describe a fluid that essentially it's not properly a transudate, it's not properly an exudate, so that is something in between. However, this term, it's a little bit misleading, because if you think modified transudate, give you the idea of a transudate that modified with time. But actually, it's not like that. I don't want to go too much into pathophysiology, but the modified transudate, it's a fluid that has a completely different pathophysiology. So the formation of the modified transudate, it's due to causes and mechanisms which are completely different from a transudate. And this is why in 2008, Stockham proposed a revised classification system of effusions, which is based on pathogenesis. And this is the classification system that many clinical pathologists, including myself, tend to use. So we have the category of the low protein transudate, the high protein transudate, then of course we have the exudate, and then we have all of a series of other or special effusions, like hemorrhagic effusion, chylus, biliary effusion, uroperitoneum, and neoplastic. And in particular, the identification of these other effusions require cytology, because that's the only way why we can discriminate, why we can differentiate this effusion from, for example, an exudate. So this was just a sort of very, very, very basic, very quick introduction to make, especially the people that are not very familiar with the topic, understanding where we are going. So I think it's time to start with the clinical cases. So I've selected for you, I don't remember now, I think 4 or 5 clinical cases. Some are much more easier and more basic. Some are a little bit more challenging, because I know that there are people that are listening at a different expertise level. And so it's time to start. The first case, it's Maisie. Maisie is a 2-year-old pit bull mix, female neutered, seen by the veterinarian because of a couple of days of anorexia, vomiting, and high temperature. As part of the diagnostic investigations that the veterinarian did, there was abdominal ultrasound, and the vet noticed abdominal fluid formation that was sampled for cytology, but also a generalized gallbladder thickening, a distension of the gallbladder, and the likely presence of some cholecystolite. So That submitted the fluid, the abdominal fluid, which we received. We did a nucleated cell count that was 28,900 cells per microliter, so significantly high. And the total protein was high as well, was 48 gram per liter. If I remember well, because the fluid had a reddish color, we also measured the manual PCV that was 6%. So I'm going to show you now a smear of the fluid, a direct smear. Obviously, it's important that I'm telling you that is direct because this means that the cellularity that you are going to see in the slide is going to be the real one, and it's not going to be artifactually high because it is a concentrated preparation. So let's have a look at this slide. Here we are with the cytology smear. As I said, it's a direct smear, so you can already appreciate at low power how the cellularity is likely going to be high, as expected from the cell count. We move obviously at a higher magnification, and I would probably say to start with a 4x. Let's go even to 10x. We can start seeing something here. We can see that the background is quite clear. We have a large number of red cells as expected because the PCV was increased and the fluid was reddish in color. And then we also have quite a lot of nucleated cells, either individually distributed or sometimes, as you can see here, forming some groups, which seems to be quite poorly cohesive. Now we can go down at this in this area, and the cells seems to be quite nicely spread, but often towards the feather edge, unless the cells are poorly preserved, but this is not the case. Often the cells are spread a little bit better, and it may be easier to appreciate the morphological feature. So we move a little bit closer to the edge, I would probably say in this area, and we go at slightly higher magnification. We go at 20 and then we move to 40. So what can we say here? We can clearly see the presence of a main population of neutrophils. I'm sure that you can recognize these being neutrophils because they have a typical U-shape, a typical segmented polylobulated nucleus, and a moderate amount of clear cytoplasm. So they look pretty much the same as the neutrophils in the peripheral blood. We then have another cell component. These are, I would say, macrophages. Sometimes mesothelial cells can also appear very similar, so it can be a bit tricky to differentiate between the two of them. And probably in this case, we have a mixture, but they are those large mononuclear cells that are here individually or that are here, and that you can see also here that they form some poorly cohesive groups. When the cell has a very vacuolated, very foamy cytoplasm, you can clearly say that it's a macrophage. When it has a sort of fringed pinkish cytoplasm, it's a mesothelial cells, but very often the morphology is kind of overlapping, so it may be difficult to differentiate between the two. But in this case, neutrophils are clearly the cell population that prevail, that you can see all around. And these, in a way, confirm that this is a exudate, I would say a neutrophilic exudate. What we should do is look for the presence of bacteria, because obviously bacteria infection is a possible differential for a neutrophilic exudate. And in order to do that, we should always move at a higher magnification. You should go to 40, 50, or 100. And that's what we are going to do right now. So we are going to move around, I would say, 60 of magnification and look. It's easier to evaluate for the presence of bacteria where the neutrophils are nicely spread, so where you can see, for example, here, the presence of the cytoplasm. And as you know, we can confirm that there is a bacterial infection if we see bacteria and if these bacteria are within the cytoplasm of neutrophils. If they're outside, they could be due to infection, but can also be contamination. And if we do not see bacteria, we can't rule out infection because cytology it's not as sensitive as culture testing for the presence of bacteria. but it's definitely worth spending some time. I always recommend to spend at least a couple of minutes. When you are a little bit experienced, you know where to go, but at the same time, you have the tendency of being a little bit too quick sometimes, and you can sometimes be wrong because of that. So spend some time. And this is a good example because if you look around, and we have seen quite a few fields up to now, there is no clear evidence of bacteria. So there is the temptation of saying, okay, it's a sterile or likely sterile exudate. However, if we look more carefully, and I took a picture before in order for you to see, there are some areas where actually we can confirm, we can see bacteria. You can see here this sort of fainted structure within the cytoplasm of neutrophils, which are likely going to be bacteria. And actually these were confirmed as bacteria because the culture testing identified the presence of E. coli. So always spend some time and look for bacteria and perform the culture in any way. If cytology is positive, it's worth to do the culture testing to define the bacteria. If it is negative, the cytology, the culture is still recommended because it's more sensitive. What we did in this case was also measuring bilirubin. There was no evidence of a bile pigment within macrophages or white bile in the background, but because of the clinical history, we were concerned. And actually, the bilirubin ended up in the fluid being significantly higher, 2 times higher than the bilirubin in the serum. And this brought us in the direction of aseptic biliary peritonitis, likely due to a perforation of the gallbladder, due to the biliary stones that were present, and also to the septic cholecystitis. I thought it was quite interesting in this case for multiple reasons, because as I said, the bacteria were not so obvious and it required some time to be identified, and also because biliary peritonitis, sometimes it presents in a way that can be identified on cytology. Other times, the only thing we see is a neutrophilic exudate, because obviously bile is a very irritant substance for the peritoneum, but we need the further testing, in this specific case, measurement of bilirubin to confirm the nature of the fluid. Unfortunately, I don't have follow-up on this case, but I have an interesting reference that I'm going to give you from a paper published in 2016 on JVIM regarding cholecystitis and cholangitis in dogs, that I think it's definitely worth reading, and it's also free download. So I will post it in the wall later on. The second case that I want to present you, it's Chloe. Chloe is a 4-year-old domestic short-hair cat with a clinical history of dyspnea and non-specific respiratory signs. That was the reason why the cat was submitted to the veterinarian. And as part of the diagnostic investigation, the vet did a chest X-ray and identified the presence of a large amount of pleural fluid that was submitted for analysis and, of course, for cytological examination. As you can see from this data, we have some numbers that are kind of similar to what we've seen before. So we have still a high cell count, so 19,500 cells per microliter, and we also have an increased total protein, 43 grams per liter. So is this an exudate, a sort of inflammatory effusion, as we have seen before? Is this something different? The only way to know that is obviously looking at the digital smear. And that's what I'm going to show you right now. Here we are. This is the direct smear of the pleural fluid from this cat. And we can already see at low power that this sample is likely to be highly cellular, which is pretty much what we were expecting, but we need to go at a higher magnification to understand better which kind of fluid we are dealing with. So now we are at 2, 4x, 10x, and already at 10x we can see that we have quite a lot of cells. They look quite different from what we have seen before, And we can have a wander around just to be sure that every area is the same as the other. I can tell you that that's the case, and most of the time with the fluids, this is what happens, except that sometimes the larger cells can be localized towards the feather edge. But in this case, the distribution of cells is quite homogeneous. So let's go at 40x and think it's the best way to evaluate the cells. So we have a clear background. As you can see, we have some red blood cells, but not as many as we have seen before. Sometimes the red cells appear quite crenated, and that most of the time it's an artifact and can be due to the contact with the anticoagulant, the EDTA, especially if there is an excess of anticoagulant. What is more interesting is noticing which cells we are dealing with. Yes, we have some neutrophils, but these represent a minority. We have some disrupted cells, some bare nuclei, as you can see here, possibly also here. We call them bare nuclei or disrupted cells when you can't distinguish nucleus and cytoplasm, and those cells, obviously, they can't be evaluated, so they should be ignored. But then we have other cells that are intact, as you can see here. And as you can see also here, and pretty much everywhere, also at this level. So let's try to understand which kind of cells we are dealing with. So they're clearly mononuclear cells, so they have one large round nucleus. They do not like neither mesothelial cells nor macrophages. Because they should have, if they were macrophages or mesothelial cells, a much higher amount of cytoplasm, so a lower nucleus to cytoplasm ratio. In this case, these cells, they are mainly composed of a very large and round nucleus. If you compare with a red cell, you can really appreciate how large these nuclei are. So they're like 3, 4, 5 times a red cell. And then the amount of cytoplasm, it's scant, sometimes moderate, and it's much more deeply blue, deeply basophilic than what we commonly see in macrophages and also in mesothelial cells. And sometimes the cytoplasm is vacuolated, but this vacuolation, it's different from the vacuolation that we have in macrophages. These vacuoles are much smaller and they're much more uniform and well distinct. Now, for those that have maybe a little bit more of experience with cytology, you will appreciate that this morphology is very much lymphoid. So the round nucleus, the eye encirration is very much lymphoid, but these cells are clearly not normal small lymphocytes because they are extremely large, as I showed you before, comparing with a red cell. So we have, in this case, a lymphoid effusion, a lymphoid proliferation, and because the vast majority of these lymphoid cells are large in size, we are definitely going into the direction of lymphoma. There are other type of effusions that can be characterized by a presence of lymphoid cells. First of all, chylus, but usually in chylus we have a prevalence of small lymphocytes admixed with macrophages. And also we have also other effusions in cats, especially in the pleural cavity. They're being called lymphocyte-rich transudate or lymphocyte-rich effusions that can be seen in association with cardiovascular disorders. But in that case, usually the cell count is lower, and again, those are usually mainly small lymphocytes. When we have lymphoid cells that are so large and they represent the main cell component, there is really nothing else than a lymphoma in the differential list. I can tell you something more of this case. The fluid was drained and the cat actually had a mediastinal mass. This mediastinal mass was also sampled for cytology and showed exactly the same population of cells. And so this was a confirmed case of mediastinal lymphoma with a secondary pleural effusion. There were no other organs involved at the time of the diagnosis, and the cat was also FELV positive, as often happens for cats that have mediastinal lymphoma. Other things that it's worth to see and to notice in this case, but not much I have to say, because the main component is really represented by these large lymphoid cells. What we often recommend in general for fluids, especially if you don't have experience, is performing a differential count of at least 100 cells. And I haven't done it in this case, but I'm sure that if you do, you will see that over 80% of these of the nucleated cells of this sample, they are actually large lymphoid cells. These lymphoma that are characterized by large lymphoid cells, they are often high-grade lymphoma, so we should expect to see of many mitotic figures. I can tell you that there were some mitosis here and there, but I can't see them right now, and I'm sure that it's going to take me forever to identify some, because when you're looking for something, you never find it. Probably after this talk, if I revise this slide, I'm sure that the mitosis is the first thing that will jump out. Are there further tests that we can do in this case? Of course there are. We can do PAR, even if it's not very useful in this case, because we already know that it's a lymphoma, so we don't need the confirmation. And PAR, it's not the recommended test for immunophenotyping. We can perform flow cytometry. Yes, we can. The problem is that we have a limited number of markers available for cats. First. Second, phenotype doesn't seem to have a prognostic value in feline lymphoma. Third, it's a mediastinal lymphoma, and large percentage of mediastinal lymphoma, they tend to be T-cell in origin. So this is one of those cases where possibly cytology might be enough. Obviously, full clinical staging, it's strongly recommended, because clinical stage, it's a prognostic factor. And then, last but not least, we have done dog, we have done the cat, and now we need to think about another species. So I have selected an exotic animal. Cytology of exotics, it's not my strongest point, but at the same time, it's one of those areas that I've been exploring recently, and I thought was interesting to share it with you. So this is a African spurred tortoise. Unfortunately, I don't have any information about the clinical signs that the animal had at the time that was referred. What I know is that the vet submitted to our attention a sample from the coelomic cavity, so essentially coelomic fluid. As you know, in reptiles and also in amphibians, we don't have a differentiation between thoracic and abdominal cavity, but there is one cavity only. This cavity contains a small amount of fluid, and during pathologic conditions, the amount of fluid can increase, like obviously in this case. So let's have a look at this slide that I think it's quite cool. It's quite cool. Here we are with the digital slide. As you can see from the way the slide looks like, this is a concentrated preparation, because you can see this is a circular area, and so the cellularity is obviously artifactually slightly higher than normal. So let's go at higher magnification and let's see what this slide is containing. So here we are. So I'm now at between 20 and 40x, and I think it's a good magnification for appreciating all the features. So what can we see? We can see that the background is clear. First of all, we don't see very many red blood cells. Remember that in reptiles, amphibians, and avians, the red blood cells are nucleated, so they have a different appearance, and if I see any, I'm going to point them out for you. And then what is interesting, we can see the presence of these structures that are present pretty much everywhere, elongated structures, and these are sperm. So you can see that there is a large amount of sperm in the background, slightly different from what we are used to see in mammals. I think you can appreciate the shape and the tail and the body quite well in this area. And then we have different cell population. We have a main cell population of large mononuclear cells that you can appreciate at this level. And these are probably cells of the macrophagic line. I'm saying that because every now and then they also exhibit cytophagia, so phagocytosis. But we also have other leukocyte type. We have some heterophils. You can see one here. You can see one here. And the heterophils, they are essentially the neutrophil counterpart, but in reptiles, and they are characterized by the presence of some sort of brick red, slightly rice-shaped granules in the cytoplasm, which can make a little bit difficult, especially if you don't have experience, differentiation with eosinophils. But these represent a minority. I think we are between 5 and 10%. We also have some small lymphocytes or possibly bare nuclei, difficult to say, but the main population is definitely represented by these large mononuclear cells. We also have some structures that are very likely going to be bacteria in the background. Sometimes it's a little bit difficult to confirm or rule out the presence of bacteria because we can have debris in the background or we can have a stain precipitate, as you can see here. It's definitely more convincing when and if we see bacteria inside the cytoplasm. And I've taken a couple of pictures before to show you. Oops, sorry. That I think will help you. You can see at this level clearly the presence of a bacteria, of a rod within the cytoplasm of this large mononuclear cell. So we can confirm that there is actually a infection. Don't think there is much else to say at this level. Let me check if there are other pictures. This is again another bacteria that you can appreciate at that level. And then again, also some disrupted cells here and there, but the main population of these large mononuclear cells, and then of course lots of sperm in the background. What I can tell you of this case is that then further imaging studies have been done, and this was confirmed to be a paratesticular cyst that clearly underwent a process of inflammation, actually of septic inflammation. But unfortunately, I don't have any additional follow-up. But I thought that was an interesting case to show you because this is definitely something that you don't see very often, or I don't see very often at least. I hope you enjoyed this case series as much as I did in putting it together. There is one take-home message that I would like you to bring home, to take home. And as you have probably noticed, all these fluids, they all had high cell count and high protein. So just by looking at the numbers, we could have classified them wrongly as exudate. But actually, with the help of cytology and knowing a little bit of the clinical context, we managed to say way more. In the first one, we managed to diagnose a biliary septic effusion. In the second one, we managed to diagnose lymphoma. And the third was a septic bacterial cellomitis. So again, the importance of cytology and how these should always be part of the fluid examination. Obviously, with this seminar, this short webinar, we couldn't cover all aspects of fluid cytology. If any of you is interested, I have a course that has just started, but you are absolutely on time for joining. That is dedicated to cytology of fluids, and where we are going to discuss into details everything that goes from the sample collection, sample preparation, and of course, sample analysis and interpretation. We're going to talk about cavitary effusion, but we're going to talk also about synovial fluid, CSF, BAL, and urine, of course. And so I think that this can be interesting not only for pathologists, but also for people that deal with internal medicine cases or with emergency and critical care, because if there is one cytology that can be urgent sometimes, I would say that is cytology of fluids, especially certain specific type of fluid cytology. There is also the possibility of enrolling into this course with the on-demand option, so starting anytime you want and doing the course at your own pace. And often people ask me, what's the difference between the two then? The course is the same, the material is the same. The difference is that if you want to do things on your own, you choose the on-demand one. If you need a little bit of extra motivation, you can enroll the traditional course. You will be into a class, and you will have some flexibility in those weeks, but there will be some work, some assignments that you need to do, and some things that you should possibly watch and read within a week of time frame. So, it's up to you what you prefer. But definitely, even if you have a busy life from a work and personal point of view, you can do this course with no problem. Just you should try to allocate a couple of hours per week, I would say, in order to complete the course on time. However, all the material will still be available for several weeks after the end of the course, and most of this material, especially the study material, the course notes, can be saved, and you can keep them for yourself, the same as for the diagnostic algorithms, for as much as you want. All these courses, they have some digital cytology built in, and this means that you will be able to navigate through the slide in the same way I did. And I think it's the best way of learning, because you will be in control of the slide. Like when you are in front of the microscope, you will choose what to look at, what to ignore, and you will then reach a diagnosis, and then you can compare that with the video solution that I'm going to provide you. So I think it's the best way, closest to reality, that we have to teach you cytology online. And after this, I want to wish you a good morning, good evening, depending on where you are and your time zone, and definitely wishing you a good weekend, and to stay tuned, because I will come back with other videos. And the only thing I'm going to ask you is if you can spread the word with your colleagues, your friends, about this group, and invite them to joining, because obviously larger our community becomes, more opportunities we will have to offer you webinars and other resources. And after this, I wish you a good day. Bye bye.